All right. Good morning, everyone. Hope that your travels to Australia have been smooth and that you are adjusting to the time zone change for those of you that had traveled far. This is the New Frontiers in Neuroimaging Meta-Analyses course. I am the course organizer, James, and one of our presenters, Angela Laird, she is currently unavailable, so I will be taking over her lectures today for this course. So we'll be going over an overview of neuroimaging meta-analyses and all the other lectures that we'll be having in this course. Just a second, I'm going to get some water. Yeah, and apologies if I pause to drink often. I am also overcoming a little bit of a cold, so thankfully there's lots of medications I can take here. So with enough pills, anything is possible. Okay, thank you all for coming. Just before I get started, for a raise of hands, how many people have ran a meta-analysis before? Okay. All right, and specifically, have you done a neuroimaging or like a coordinate-based meta-analysis? Who has done that before? Okay, nice. So with that in mind, I'll be able to tailor the lecture on what to spend the most time on. Okay. So I'll say that I have no disclosures, and for this lecture, we're going to work on understanding the role of meta-analyses in neuroimaging and the broad families of methods which exist under neuroimaging meta-analyses. So specifically, we'll be explaining the limitations of single studies and how that relates to the reproducibility crisis, which many people have already have heard of or have dealt with in the 2000 teens. We're going to explain the main uses of meta-analyses and outline some of the broad family of methods. So this is the Neurosynth team. Throughout these lectures, there are different ecosystem of tools that we have access to. So this is one ecosystem of tools built by these people, including me, for Neurosynth, and we'll be introduced to Neurosynth Compose in one of the later lectures as well. So again, goals for today: learn the motivations for neuroimaging meta-analyses, familiarize yourself with the landscape of tools and the methods for meta-analysis, understand the common pitfalls in statistical inference and meta-analysis, and learn to critically evaluate results. And we'll be giving you some hands-on experience on conducting neuroimaging meta-analysis using some of the best practices for preferred reporting in systematic meta-analyses or in systematic reviews and meta-analyses, otherwise known as PRISMA. So in this and all the subsequent lectures, we are primarily looking at fMRI meta-analyses, but this can be extended into volume-based morphometry and PET and many other modalities that report coordinates as one of their outcome measures. And then going over the lectures, overview of motivations, which is the one that you're currently listening to, then coordinate-based meta-analyses, then LLMs for metadata enrichment, and running a coordinate-based meta-analysis using NeuroSynth Compose. And then we'll be talking about the past, present, and future of image-based meta-analyses, as well as the image-based meta-analyses methods and algorithms that you have access to, and running an image-based meta-analysis using data from NeuroVault, which will introduce NeuroVault if you haven't heard of it before. Okay. So taking a step back, why are we trying to understand how are we using these brain images to understand the human brain? We are trying to map regions associated with a particular mental state, such as which brain region signal to pain. And to identify the function of a brain region, such as what mental processes does the dorsolateral prefrontal cortex support, and characterize the role of brain networks. What are distinct subcomponents of the default network, and what do they do? And we want to be able to do this not just within healthy individuals, but also within people of varying populations, whether they're diagnosed with schizophrenia or depression. We want to be able to generalize across healthy participants and see if those same processes also generalize across people with disorders. So moving from concepts to brain maps, it looks like there's a little... we start out with a concept, such as cognitive control, and when we move on to how we want to measure that, we can look into the specific tasks and how we measure cognitive control or conflict adaptation. And we can look at Stroop or the go-no-go task, which are standard tasks that purportedly measure this idea of conflict adaptation. And then we do that measurement in 20 participants, and we run a contrast to say, okay, then this brain region, the dorsolateral prefrontal cortex, is associated with conflict adaptation. We found out that that is not enough. Just grabbing 20 people that are accessible through your college or university and running an experiment just with those 20 participants does not result in very reproducible results. And one of the hallmark studies that keeps coming back over and over again, or keeps getting brought up over and over again in these hallowed halls, is the dead salmon study. And I guess raise your hand if you don't know about the salmon study. Okay, nice. So many people may know about the salmon study, but in case you don't, the dead salmon study was done a little bit as a tongue-in-cheek way of looking at the error of not performing multiple comparisons correction in fMRI. So they literally put a dead salmon inside an MRI scanner and measured its activity over time. And when they ran the analysis on the data from the dead salmon, they found significant voxels of actual data saying that the dead salmon actually presented new results and activity inside of its brain, which does not make sense because the dead salmon does not have any activity. So that was a warning sign in the field of neuroimaging to suggest that we need to have corrections, and we need to have corrections for all of the comparisons that we're running in these analyses, because the human brain is represented by 100,000 voxels, and if we just run an independent test on each one of those, just by random chance, there's going to be a few that are going to pop up and say that they are significant. Okay. So that put us in a bit of a bind of suggesting, okay, we are running these analyses with not a lot of subjects and using methods that may be duplicitous in how well they help correct our statistical results. But I think that this also gave us an opportunity. So the opportunity is being able to combine data sets from different universities and different studies. And finding all of this, dealing with this reproducibility allows us to really look into sharing data sets and sharing results with other labs and other universities. And so there's been multiple efforts that have started up over the years in order to try to create these opportunities to share data and make it easier for individual researchers to aggregate information across all of these different studies. And so this isn't necessarily a new thing either. Even way before the dead salmon study, people have been interested in trying to combine information across these maps that we've extracted from across these brain maps. And it was very rudimentary, where it was essentially trying to align the brains with each other using mostly eyesight and some and diligence and time. But again, what are these limitations of these individual neuroimaging studies? Is that we have small sample sizes, when especially compared to other fields of cognitive and social science. And another particular limitation is we aren't looking at neural activity itself. We're looking at a proxy of neural activity. So, namely, we are looking at the blood flow that is feeding the neurons and the brain regions that are being active. So, there are a host of other confounds to be aware of. And because MRI is rather expensive, to perform, we have these small sample sizes, and it's really difficult to have these other follow-up and confirmational studies because there's simply just not enough resources to conduct all of these additional studies. And when we talked about brain mapping earlier, when we're going from, say, conflict adaptation to measuring it with the Stroop task, what we're trying to say is something about conflict adaptation. We don't necessarily care about the idiosyncrasies of the Stroop task or the go, no-go task. What we're trying to say is these brain regions represent something fundamental about conflict adaptation. But all we have access to are the particular contrasts that we developed through these specific tasks that we have the participants complete while within inside the scanner. And with all these limitations, we are also being over-encumbered with the number of publications that we have within the fMRI literature. So we are able to have access to all of this literature, and even though this is a number, we do have many more papers that we can analyze across. Sample size is still not sufficient, and we need meta-analysis in order to synthesize all the findings that we have in this literature, because it's otherwise impossible for any individual to read all of the literature that's accessible through here. So now that I mentioned meta-analysis, what exactly is it? A meta-analysis combines findings from individual studies to build consensus of an estimate of brain activity for a given set of analyses. So if we have a set of studies, which I call a study set, each of those is a study, and each of those studies reports coordinates in these tables, and we can map those coordinates onto an actual brain. And so if we, in this particular example, we have 162 studies that are about emotion, and we can identify the areas in which there's the most overlap for the between all of these studies. And so the most bright regions are yellow, and that means across the 162 studies, those were the areas in which the most coordinates were reported. And thankfully, within the field of neuroimaging, we have created standard templates in which to report these coordinates. So we have the Montreal's Neurological Institute. They have an M&I space that they've worked on to define, as well as there's Talerac that was before M&I was created. So again, here's a representation of these coordinates that we can see within the brain. And what we are looking for across all these coordinates is where they converge. So if we're just looking at all of the coordinates individually, it looks like, well, this process happens all over the brain. But when we look at the ones in which it converges, in which there's the highest density of coordinates, then we can start making conclusions about which regions converge. So we're not doing a typical effect size meta-analysis in which we are looking at the strength of the effect. We are looking at a location effects meta-analysis. So which regions of the brain are most reported. And so how we do this is we take each coordinate and we essentially smear it by convolving, we say, foci with the spatial kernel to create study-specific modeled activation maps. So we'll take a coordinate, then we'll smear it a little bit, and then we can combine those modeled activation maps into a sample-wise map. And then we can compare that to a null distribution to determine the voxel-wise statistical significance. And so this has also been going on for a while in order to determine meta-analyses. Since in the very early days, we've been reporting coordinates in these papers. And as we wanted to combine across these studies, a lot of work by Peter Fox and others have created methods to integrate brain maps and to go beyond the single study and combine multiple studies, and eventually created their own database called, doing as a framework called BrainMap. And this has continued to look at other types of studies to differentiate the functionality of specific regions. And with Peter Fox and company, they've developed a website called brainmap.org, in which it is a finely curated list of studies and their coordinates that people have used in order to conduct their own meta-analyses. So again, here's some more examples of other meta-analyses, and then at the very end here, I'm going to talk about the NeuroSense database. That was another database that was created. Instead of being finely curated, this one was roughly scraped from websites and using information that could be scraped from published web pages. They use this to create automatic meta-analyses. So what it lacks in quality, it made up for in scale relative to the brain map, database. So with the neuroimaging meta-analyses, we have what we call coordinate-based meta-analysis. So again, we convolve foci with a spatial kernel to produce study-specific maps. We combine those MA maps into a sample-wise map, and then we compare that to a null distribution. So within that, we have multiple methods, such as activation likelihood estimation, ALE. There's kernel density analysis, which has been superseded by multi-level kernel density analysis, MKDA. There's also ways of measuring coactivation, so specific coactivation likelihood estimation, as well as seed-based demapping, as well as MKDA chi-squared, which is an extension off of the multi-level kernel density analysis method, which allows one to compare their specific results to ideally another set of studies to see where your set of studies uniquely contributes to the results as opposed compared to a prior. And coordinate-based is well and good, but the other method, and the method in which we would all like to head towards, based on how well we're sharing our data, is image-based meta-analysis. So within image-based meta-analysis, we got mixed effects, general linear models, fixed effects, general linear models, Fishers, IBMA, Stauffers, and Z permutation. And we'll be covering some of these in more detail in some of the later lectures. And where do we get these maps, and where can you get these maps, and where can you share your own statistical maps? That would be on NeuroVault. NeuroVault is been available since 2015 or 2014, and has been the one-stop shop for uploading all of your statistical maps, your atlases, and other useful brain images. So as a resource, you can upload maps, and you can download existing maps on here for your own research interests. And our goal, here for the field of neuroimaging and in science in general is to have meta-analyses be a part of the research cycle that is easy and just happens as a part of doing research. So we want to make meta-analyses so accessible. It's part of the common research practice rather than a specialized product. But the meta-analyses are extremely time intensive. So in order to conduct a meta-analysis, you have to find your studies, you have to curate all the studies, you have to extract information from the studies, then annotate them with whatever features that you need to annotate them with, specify how you wanted to model all of these studies, and finally execute the meta-analysis. And so how do we choose what goes into a meta-analysis? There's a number of choices that one needs to make, and again, this is the preferred reporting items for systematic reviews and meta-analyses, or PRISMA. And you will go through items that seem relevant to your research question, as well as determining what needs to screen for relevance, and then seeing if the articles actually contain the information that's needed for the meta-analysis. So you see if they actually do they report coordinates, and do they have the other pieces of metadata that describe what you need for your meta-analysis. And the main outcome, or the idea, or the main, some of the main things, is that the assumptions, the input to meta-analysis represent a common process or state. So whether that means to you, what we talked about earlier for trying to measure conflict adaptation, does that mean we're going to include all these different tasks that say that they measure conflict adaptation, or are we just going to choose one? And what does that bias in terms of specificity versus power? And from Simon Eickhoff and colleagues, they've determined about 17 to 20 experiments is what's needed to have a good meta-analysis where you don't have a small number of studies driving the effect. Okay. So we covered a lot. I want to say individual, but the overview is individual fMRI studies remain significantly underpowered. We have many false negatives as well as false positives and inflated effect sizes. Meta-analyses are readily available solution for synthesizing literature, and coordinate-based meta-analyses is currently the most common method due to data availability, but it does have its limitations. IBMA is superior, but is unpopular due to lack of data, but we'll be talking about this more when we touch on NeuroVault. And the most critical scientific aspect of a meta-analysis is the inclusion criteria. So this is the part in which you really have to think carefully and be systematic and transparent about why you're including a certain study or why you're excluding a certain study. All right, thank you. Are there any questions for this lecture?